Hi friends, I'm Dr. Divya. Welcome to classes by Lab Doctor. And in today's class, let us discuss about the sperm motility. This sperm motility is the one which will help the sperms to go and meet the egg and fertilize it. So it becomes a very important factor when it comes to male fertility. So it is very important that we correctly identify the motility, we grade it correctly so that it will help the patient as well as the doctor to treat if needed in case of any abnormalities. कितना सैंपल असेसमेंट करना है, सेमेन एनालिसिस है, उससे भी इम्पॉर्टेंट होता है करेक्ट सैंपल कलेक्शन. ये बहुत इम्पॉर्टेंट है when we are trying to assess the semen motility. And also ये whatever sample is there, it has to be liquefied within 30 minutes and we have to proceed to see the motility only after the sample is liquefied. अगर sample liquefied नहीं है and if we try to assess the motility, we might give a wrong result. Suppose sample does not liquefy, then we will have to incubate it for 37 degree for another 30 minutes and then only the analysis has to be started. Also remember the motility of this sperms are temperature dependent. That is why we will have to standardize the temperature during the motility assessment. Actually, we are supposed to use a microscope that is equipped with a temperature controlled object stage, microscope slides and cover slips that are pre-warmed, and the sample is also warmed at 37 degree before assessment, because all these things are required to correctly assess the motility, because the sperm motility is temperature dependent. So now there are different grades of motility which the WHO classifies. One is rapidly progressive. So the name suggests that rapidly it is progressing. So the sperms are moving and it is moving rapidly. So it is moving either in a linear fashion or in large circles, but it is moving very fast from one point to other point. Second one is slowly progressive. So here also the sperms are moving, but they are moving slowly. So they are moving either in linearly or they are moving in large circle, but the speed is less. So that is slowly progressive motility. Third one is non-progressive. These sperms are not moving. And suppose what is moving, if at all they are moving, their flagella is moving or the head is only displaced less than 5 micrometer. So that sperms are called as non-progressive sperms. And last one would be immotile sperms. The sperms are not at all moving. In this slide you can see that there are sperms which are rapidly progressing. So they are moving very quickly and they are moving in either linearly or they are moving in large circles and they are traveling from one point to other point. You can see that they are moving from one point to other point very quickly. The distance is more than half the tail length. Within 1 second they are moving from one point to other point. Here you will see a slowly progressive sperm. It is moving, definitely it is moving in a circle or in a linear way, and but the distance which is traveled from one end to other end is less than 25 micrometer or half of the tail length. The distance is covered, but it is moving slowly, it is progressing. So this is slowly progressive category. Here you can see some sperms, they are totally immobile, they are just not moving. So these are called as the immotile sperms. Here you can see some sperms which are not moving, or they are non-progressive sperms. So here you can see only their tails are moving, the flagellas are moving, and the head is not moving. Even if the head is moving, it is less than 5 micrometer the displacement. So these are the non-progressive sperms, only the tail or the flagella being moving and they are not progressing at all. This different categories of sperm motilities have to be mentioned in the report. So progressively motile sperms are the one which are moving very actively. They are either moving in a linear direction or in large circle regardless of the speed. that is progressive motility, and that represent the quality of the sperms. The sperms which are progressively motile are the ones which will be able to fertilize. And this progressive motility, it could be rapid or it could be slow. Then you have another category called as non-progressive. Here what happens is, these sperms, they are swimming, but they are swimming in very small circle and only the flagella is moving, or hardly the head is being displaced. So those are called as non-progressive. They are not able to progress. They are not able to move in a in a linear fashion or in large circles. Those are non-progressive. And third would be immotile. There is no movement at all. So if the motility, if it comes less than 50%, then we will have to do the other test like sperm vitality test, etc. Suppose if the motility is 0, then we might have to do the pentoxyphylline stimulation test. So sometimes we also tell it as grade A, grade B, grade C, grade D. So grade A is nothing but the fastly progressive sperms. Grade B is the slowly progressive sperms. Grade C is the non-progressive sperms, and grade D are the immotile sperms. This is a semen analysis report where you can see they have graded the rapidly progressive, slowly progressive, the total progressive sperms, that is rapidly plus slowly progressive, the non-progressive sperms, and immotile sperms. How do we assess the sperm motility? So you will have to mix the semen sample well. Then you have to transfer around 10 microliter of the semen sample on a clean slide, and you have to cover it with a cover slip around 22 millimeter depth of the cover slip. So when you do a wet preparation, you can examine it at under 40x, and you can see that you avoid the edges. And whenever you are counting the sperms, you should use the counter. Evaluate at least 200 sperms in each replicate. Okay, so high sperm concentration, if it is there, you can select random fields. Suppose agar sperm concentration low hi hai, then you will have to screen the entire slide. You will have to calculate the average, and then you will also have to find the difference between the two percentages, and according to the acceptability difference you will have to give the report. So this is the wet smear which is prepared, and this is you have to examine it under the microscope. So this is an area what you can see. Avoid this area at the edges. Always count the complete sperm. No pinhead sperms, no only tails or no only heads have to be counted. It is the entire sperm which has to be taken. And here you can see they have counted around 200 sperms, and they have written which are the progressively motile sperm, non-progressive sperms, and the immotile sperm. So you will have to take the count. Suppose now you did suppose you did the counting of 200 sperms, and in the other replicate you got 68%, and in one slide you got 60%, and in the non-progressive one slide you got 10%, other you have got 5%, and the immotile in one slide there is 30%, and other it is 27%. So the average difference is around 10 is fine. Suppose if the difference is more than 10, then we might have to do another replicate study. So here you have to take an average because the difference is less than 10. So here we will have report this progressive motility as 64%, non-progressive motility as around 8%, the average, and the immotile as the average would be around 28%. So it is very important that you do the sperm motility in replicate of 200 sperms and then calculate the average. The difference should not be more than 10%. Important thing is that you will have to assess only the intact sperms. Score the 3 categories in the same field until you count 200 sperms. So what is the normal parameter? The normal parameter would be the progressively motile sperms have to be at least 32%. This is the lower reference limit. And the total motility is progressive as well as non-progressive. That should be at least 40%. So total motility, that is progressive and non-progressive should be at least 40%, and the progressive motility what we see should be at least 32%. Sometimes if the sample is not liquefied, suppose if it is highly viscous, you have made the analysis very late, then you will get a wrong motility report. So that is all about sperm motility and the different grades of sperm motility. In the next class, we will see what all can go wrong with the sperm motility and how do we treat or how do we improve the sperm motility. Thank you so much for watching the video. Please like, share and subscribe the channel. Thank you.